Looks good.
Okay, terrific. So thanks so much for the opportunity to speak to you. I'm really excited to be here and tell you a little bit about some work that's been going on in my lab for quite a while now related to barcoding for species identification. So we're all pretty familiar with the concept of barcoding in microbiology, which is this use of small, short, informative sequences that we can amplify with PCR and sequence. And this has been done for a long time with individual isolates, and now with the advent of advanced sequencing technologies, it's a common method to approach microbiome profiling, in which we take some unknown sample, we amplify this informative target sequence, and then compare the amplicon sequences for species identification. And metagenomics has been an amazing addition to our repertoire of things, but barcoding remains a really good go-to method for rapid profiling of communities. So that informative sequence that's used, for the most part, has been the 16S ribosomal RNA gene. It's widely used for identification, and there are universal primers for amplifying different variable regions of that target, massive databases of reference information. But the one sort of potential vulnerability of using that technique is that often resolution can be limited, and frequently things are only reported to the genus level because species identification isn't possible. So in my lab for a while, and from people actually before me started this work, exploring an alternative target for this kind of work, and that's the chaperonin 60 gene. It's also called cpn60. It encodes the 60 kilodalton chaperonin protein. This is found in bacteria, but also in eukaryotes and even in some archaea. And if you don't immediately recognize the name, you might recognize some of its synonyms. It's also known as HSP60 or groEL, particularly in E. coli. So why this target? The reason why we're using it is because it meets all the criteria for a sequence barcode. It's universal. It's found in prokaryotes and eukaryotes. It is accessible with universal PCR primers. There is a reference database, a curated reference database called cpnDB for the sequence data. And then perhaps most importantly, it's very informative. And in fact, it's more informative in a lot of cases than ribosomal RNA sequences are. So interspecies differences tend to be much larger with cpn60 sequences, which means it's easier to tell species apart. And as a bonus, it's also usually a single copy in the genome. So it makes an intriguing target for quantification methods as well. So over the years since the original publication of the universal primer approach, this target's been used by groups, my own group and others, for a variety of purposes, including beginning with identification of clinical isolates and then moving on to phylogenetic analysis, new genus and species designations. It's turned out to be a really excellent predictor of whole genome sequence similarity, which has led us to being able to differentiate between strains and ecotypes within species. And we exploit it a lot in our lab for as a diagnostic target, using especially real-time PCR. And then finally, what I wanted to focus on today, which is microbiome profiling or determining taxonomic composition. So this has been done with standard amplicon sequencing and also by some colleagues of ours using targeted methods such as capture-seq. So this workflow has become really common for us, and we've published this protocol. It's out there for you if you're interested, of going from a microbiome sample through barcode amplification and sequencing and comparison to our reference database to generate these microbiome profiles. But as the studies have gotten larger, sample numbers are higher, the amount of data we're generating is more, we started to come back to asking over the past couple of years some really important basic questions, one of which is how much of the barcode sequence do we actually need? The whole thing is about 555 bases, but we know that sequencing costs money, and the less of it we need, the better. So we wanted to figure out how much we needed. And then we also wanted to reconsider the ways in which we're comparing the sequences we generate to our reference database. Because again, as the data sets get larger, things slow down and it becomes more computationally intensive. So the first question, how much of the barcode do we need for identification, was addressed by a former student of mine, Sarah Van Kuren. And we often use now the formation of amplicon sequence variants to describe the unique sequences in a microbiome. And we know that in order to do that, you have to truncate all of the reads at a consistent uniform length. And we know there's a trade-off between sequence length and the amount of data that passes filtering. So if you require a longer sequence, you're going to retain less of your data. So Sarah took several 100,000 sequence reads, all high quality at 300 base pairs from the vaginal microbiome, a community that we work on a lot. And she used that as input for variant calling. And what she found, as you can see on the right, the purple bars, as you increased the truncation length, there really wasn't any major gain in the number of unique sequences that were detected. So that was good. And more importantly, if you look at the right-hand axis, we looked at the percent of the ASV that only found one best match in the database. So it was an unambiguous identification. And once we got to 150 base pairs, there was really no gain in that. So we concluded from this that 150 base pairs of the target is sufficient to get unambiguous and usually species level identification. So how do we then compare these 150 base pair queries to our database? Historically, we've been using alignment-based methods, combination of BLAST and Smith-Waterman alignments. And the result from that, as you'll see on the right, an example with 2 queries there, is a percent identity to the best match in the database, which in the case of query 1 here is great. It's 100% identical to Helicobacter canis, so we would feel confident about that identification. But for query 2, there is no perfect match, and so we're left with a best match, which means when you're reporting these hits out, there's a lot of interpretation that has to be done, and it can be a little bit misleading. The other thing is the database and the query set gets larger. Alignment methods are computationally intense, and so things can get quite slow. So we wanted the ability to do variable rank classification and to have a confidence score for these identifications. And so we turned our attention to looking at classifiers, which have been widely used for 16S identification and metagenomic sequence identification. So Qingyi Ren in my lab trained a couple of popular classifiers, the RDP classifier and the QIIME2 feature classifier, on cpn60 data just to see how it would perform. And she compared those results to the alignment method we were using and tested that out on several human microbiome data sets. So what she did was she took the assembled ASV sequences from 3 different microbiomes, vagina, stool, and saliva. They were all done at 150 base pairs. And then she did our alignment method to generate the nearest neighbor species, and then also tested the 2 classifiers, looking at the ability to classify to the species level. So what she found in general, first was that the classifier species results were very similar to the alignment results. In fact, they gave the same result for over 90% of the ASV. And we got nearly identical results from these 2 particular classifiers. And if you look at the pie charts on the right, the first column there, the proportion of ASVs that were classified to the species level is shown in purple. And it's, by far the majority of sequences. But we know, if you take a look at the histogram on the left, that we always in a microbiome have some ASVs that are present at very high abundance and some that are extremely rare. And so if we look at the proportion of the microbiome that the sequences represent, that's in the right-hand column of pie charts there, showing us that we, in fact, are classifying the vast majority of the microbiome to the species level using this approach, with 150 base pairs from the 5' end of the cpn60 barcode. So in summary, what I've showed you is that 150 base pairs or more of the barcode provides species level resolution. It's generally better than you'll find with any of the variable regions in 16S, and that the amplicon sequencing protocol is available in Protocol Exchange. And then the other thing I talked about was that the classifiers are compatible with cpn60 classification. We've looked at 2 so far, and we're in the process of examining others to see if we can even improve that performance. So I'll just wrap up with thanking my group and pointing you to our coordinates on the internet. If you're interested in the protocols or have any questions, we're always happy to offer advice and support if you're interested in adopting some of these methods. So thanks very much, and I look forward to your questions.